Have you ever wondered how cells duplicate themselves? Every single cell in your body undergoes a process called the cell cycle. This cycle, akin to a cellular dance, is a carefully coordinated sequence of events that allows cells to grow, duplicate their DNA, and ultimately divide into two. The cell cycle is divided into distinct phases, each with its own unique functions and purposes. It begins with a resting phase called interphase, which is then followed by the mitosis phase where the cell divides. But nestled within interphase, there are two other critical stages: the synthesis phase and gap2 phase, where the magic of DNA replication and preparation for cell division happen respectively. These phases are not just sequential steps on a cellular assembly line. They represent a carefully orchestrated process, controlled by a complex network of signals and checkpoints, ensuring that each new cell is a perfect copy of its predecessor. So, stay tuned as we delve deeper into these fascinating phases of the cell cycle. Before a cell divides, it goes through a period known as interphase. This period is no ordinary rest. It's a bustling hub of activity, preparing the cell for the important journey ahead. Interphase is a phase of growth and preparation, divided into three subphases: the G1 phase, the S phase, and the G2 phase. In the G1 phase, the cell grows and synthesizes proteins, setting the stage for DNA replication. The S phase is where the magic happens, the DNA replication. It's a meticulous process where each strand of DNA is copied, ensuring genetic information is preserved. The G2 phase is the final preparation for cell division. Here, the cell checks for DNA damage from replication and makes necessary repairs. It's like a final quality check before the cell division. Each of these phases plays a critical role in ensuring that the cell is ready and prepared for the next big step. Interphase is crucial for preparing the cell for the next big step, division. The synthesis phase, or S phase, is where the magic really starts to happen. It's a key part of the cell cycle's interphase, during which DNA replication and the formation of histone proteins occur. These processes are crucial for cell function and are meticulously regulated to ensure precision. Histone proteins are particularly significant during this phase. Why? Well, these proteins are essential for forming nucleosomes, which are needed by the newly replicated DNA molecules. The S phase also incorporates a regulatory checkpoint, the G1/S checkpoint, which only allows cells with sufficient nutrients and undamaged DNA to proceed. As this phase unfolds, the DNA content within the cell doubles, but the chromosome count remains the same. This is because chromosome division isn't on the agenda just yet. The S phase ensures that DNA synthesis happens before the M phase and with exactitude. At the end of the S phase, each chromosome of the cell has double the amount of DNA with a double set of genes. Following the S phase, the cell moves into the gap2 or G2 phase. This phase, like a diligent worker preparing for a big presentation, is the cell's time to gather nutrients and release proteins. It's all about preparing for the grand event of mitosis, the M phase. But don't be fooled, it's not all about preparation. The G2 phase also plays the role of a quality control officer, meticulously checking for DNA damage that might have occurred during replication. This quality check ensures that the cell is fit and ready to undergo division. Now, you might find it interesting that some cells, especially those that divide rapidly, like our skin cells, may skip this phase. They go straight from DNA replication into the mitotic phase. However, for most cells, the G2 phase is an essential checkpoint. It's like a gatekeeper, preventing mutations and DNA damage from causing excessive cell proliferation. The G2 phase plays a pivotal role in ensuring the cell is in proper condition to undergo division. Finally, we arrive at the mitosis or M phase, the grand finale of the cell cycle. This is where all the preparation and meticulous planning come to fruition. It's during this phase that the cell splits into two identical daughter cells, each carrying a full set of chromosomes identical to the parent cell. The M phase is a complex and intricate dance, divided into four subphases: prophase, metaphase, anaphase, and telophase. Let's delve into these subphases to understand the process better. First up, we have prophase. This is the beginning of the mitotic phase where the DNA and proteins in the nucleus start to condense into tightly coiled chromosomes. The nuclear envelope begins to disintegrate, making way for the spindle fibers to attach to the chromosomes. Next, we transition to metaphase. The chromosomes, now attached to the spindle fibers, align themselves along an imaginary line in the center of the cell, known as the metaphase plate. This alignment is crucial for the even distribution of genetic material to the daughter cells. Following metaphase, we enter anaphase. Anaphase is like the grand reveal of a magic trick. The sister chromatids, which make up the chromosomes, separate and move towards opposite ends of the cell. This movement ensures each daughter cell receives an identical set of chromosomes. Lastly, we come to telophase. In this final act, a nuclear envelope forms around each set of chromosomes, creating two separate nuclei within the cell. The chromosomes then begin to unwind, reverting back to their relaxed, string-like form. But the show is not over yet. The cell now proceeds to cytokinesis, a process not technically part of mitosis, but often associated with it. During cytokinesis, the cell itself divides in two, marking the birth of two identical daughter cells. And thus, through the careful orchestration of these phases, a cell successfully duplicates itself. The cell cycle is a complex but fascinating process that happens countless times in our bodies every day. It begins with the cell's resting phase, known as interphase, where the cell prepares for division. The synthesis phase, or S phase, follows, where DNA replication and histone protein formation occur. These processes are vital as they ensure precision in DNA synthesis and protein production. Next comes the gap-2 phase, or G2, where the cell gathers nutrients and releases proteins, gearing up for the mitosis phase. Here, the cell checks for any DNA damage that might have occurred during replication, ensuring that it is in prime condition for division. If any damage or insufficient nutrients are detected, the cell remains in the G2 phase, not progressing to cell division. Finally, the mitosis phase, or M phase, takes place. This is where the magic happens, and the cell divides to form two identical cells. So the next time you look in the mirror, remember the billions of cells that have gone through this incredible process to make you who you are today.